So we started the discussion of second messengers, and in the last class, the properties of second messengers was the discussion that we had. In that, the next following lectures will be the examples in detail we are going to discuss in the second messengers. So last class we were discussing that second messengers of two types, soluble second messengers and some messengers will be associated with the membrane. So membrane associated second messengers, these are the lipid in nature because they are associated with the membrane, which is the lipid layer basically. So lipid based signal transductions are associated with such type of second messengers. And soluble, they are polar in nature. So soluble means water soluble, suppose. So in the cytosol, much of the content is nothing but water. So water soluble means they are polar. So polar second messengers are migrating to the target site by simple diffusion, right? So now in detail with the examples we have to discuss. So CAMP is one of the soluble type of second messenger. So CAMP, calcium are soluble type of second messengers. Last class we started the brief introduction about CAMP and then signaling functions of CAMP and CGMP. Today we are going to discuss. And then the another important second messenger like CAMP, CGMP and guanylyl cyclase. So in case of CAMP, adenylyl cyclase is the one which is responsible for the formation of CAMP from ATP, whereas in case of CGMP it is guanylyl cyclase. Then targets of the CGMP. That's all for today's discussion that we are going to have. So cyclic AMP, as we discussed, is basically a soluble type of second messenger, very important second messenger in the cell. Now you understood that second messengers are like mediators which mediate the upstream signal transduction with that of the downstream effector molecules. So the CAMP is a very important second messenger, soluble type of second messenger that we discussed, and it is present in the cytosol. And the formation of CAMP is taking place from the ATP by the action of adenylyl cyclase, which is denoted as AC. And then degradation of the CAMP is because of the enzyme known as phosphodiesterase. This is indicated as PDE. So the concentration maintenance of CAMP in the cytosol is very important. That gives the condition of the cell, under which condition that particular cell is present. So based on that, the signal transduction pathway, respective signal transduction pathways and cellular events will be associated. So this concentration of CAMP is basically mediated by these two enzymes, synthesizing enzymes and degradation enzymes. Okay, so they both responsible for proper maintenance of the CAMP levels in the cytosol. So the CAMP can influence many of the cellular physiological events like it is associated with gluconeogenesis, glycolysis, lipogenesis, muscle contraction, membrane secretion, learning process, ion transport, differentiation, growth control, and apoptosis. So all these physiological events are associated with CAMP levels in the cell. That's how CAMP concentration and its maintenance in the cytosol is very important regulation point. So both the enzymatic activity in that way, adenylyl cyclase and phosphodiesterase, they function in a cooperative manner so that the maintenance of CAMP is depending on spatiotemporal, that is at the particular location and at particular time. So important feature of CAMP is another important feature as far as CAMP signal transduction is concerned, that is co-localization. So CAMP co-localization where the target is present, the CAMP target is present, there CAMP has to has to migrate at that particular place. So example, adenylyl cyclase, that is the enzyme which is responsible for converting ATP to CAMP. CAMP formation or CAMP synthesizing enzyme, adenylyl cyclase. Phosphodiesterase breakdown enzyme. After its function, cyclic AMP has to be acted upon by phosphodiesterase. And protein kinase A. Protein kinase A is the substrate, the downstream effector of CAMP. All these have been found to co-localize at the same subcellular sites, allowing for precise control of CAMP formation, degradation, and target selectivity. Understood? So, at particular localization, all these will be present, and that is very important for the function to exhibit. So next is the signaling function of this CAMP. How this CAMP signal transduction pathways. So you need to practice, as I said. Okay. So all these diagrams you have to keep practicing. You see now the pathways here. I given the depiction like A and B. So linear pathways. So you have signal transduction pathways either linear or cyclic. Now these are linear pathways. So this is pathway like glycolysis and gluconeogenesis, for example, pathway A and pathway B. Now these two pathways can be interconnected. The metabolites of one pathway can form the metabolites of another pathway. This type of interconnections in between, intermediary connections between two pathways are known as signaling crosstalk. Crosstalk signaling. Okay, so this term you need to memorize. So crosstalk signaling refers to the pathways which will interconnect the two major pathways. Understood? So those are crosstalk pathways. So such type of crosstalk you can see here. This is one pathway, this is another pathway, and these two are connected by some common substrates. So here the discussion is for CAMP. Those CGMP because they are the crosstalk mediators. That's why it is here. CGMP is next topic. But in CAMP, in the question if you get, you have to write this diagram. So ATP, what we discussed, ATP is converted or ATP is the source for formation of CAMP by the action of adenylyl cyclase. And this adenylyl cyclase is getting activated. This is present in the membrane, right? It is associated with membrane, this enzyme. And this is by the heterotrimer. This heterotrimer, you remember, that is heterotrimer is the G protein which is bound to the G alpha is a G protein, G domain, and it is associated with G beta gamma. G alpha beta gamma is a heterotrimeric protein which is associated with G protein coupled receptors. And these proteins, along with calcium, can activate adenylyl cyclase. And this activated adenylyl cyclase converts ATP to form CAMP. And this CAMP, as we discussed earlier, depending on the condition and the time and the location, also depending on the requirement for that particular pathway, the levels will be maintained how much it is required by the degradation enzyme, that is phosphodiesterase. This acts on CAMP converting into AMP. This breaks the cyclic structure of the cyclic AMP, and that's how CAMP levels will be regulated by the action of PDE and adenylyl cyclase. So both are reversible reactions. Okay. So this CAMP has got three major targets in the cell. So EPAC, this is one of the GF protein. This is an EPAC here. And one homework for you. You have to tell tomorrow what is the expansion of this word EPAC. Okay. So what is EPAC? You need to tell tomorrow. PKA, you know already, right? PKA is very important kinase that is associated with CAMP. This is also known as CAMP dependent protein kinase. PKA is also known as CAMP dependent protein kinase. Already we discussed PKA, PKB, PKC. Okay, PKC. So here, this PKA is the major target of CAMP. Then there are cyclic nucleotide and gated ion channels. These are also affected by this CAMP. And these are the common targets even for the activated CGMP. Okay. And there are so many similarities if you see CAMP and CGMP. In this case, nitric oxide is the another second messenger which is responsible for activation of guanylyl cyclase. So guanylyl cyclase, like adenylyl cyclase, is responsible for formation of CGMP from GTP. And the reverse of this, same like in CAMP to AMP, mediated by phosphodiesterases. Those are the same enzyme which will break the cyclic structure of GMP also. So CGMP is converted to GMP by the action of PD. Understood? So these two again here reversible reaction. Instead of adenylyl, here it is guanylyl cyclase. Understood? So that way CGMP is formed. And in case of CGMP, the major target is PKG. So how many PKs we discuss? PKA, PKB, PKC, and PKG. So PKG today we are going to discuss because this is the target of CGMP, which is an important second messenger in the cell, like that of CAMP. Understood this all? So this activated cation channels by both CAMP, CGMP. You see that in calcium-mediated neuronal impulses, so calcium signal, electrical signals that is in the brain, okay, in the brain and nervous system, these signal transductions are mediated even in the visual cycle, okay, in the visual CGMP role in the visual cycle. So this is mediated by through the targets of cation channels, and that will open for the cations calcium, and that is the important basis of vision. Understood? So that is all about CAMP signaling and its function. So here the EPAC is one more the effector, and that mediates the effects through MAP kinase. MAP kinase, you know, right? So MAP kinases are important signal transduction pathways in the cell. MAP kinase, what is the abbreviation? Earlier it was thought that it is microtubule associated protein kinases. Microtubules, do you know? If you don't know, you have to tell me tomorrow what is microtubule. Because they found initially these kinases are mainly associated with microtubules. That's why they call microtubule associated protein kinases. The same kinases afterwards, they renamed it as mitogen activated protein kinases. What is a mitogen? Mitogen is a growth factor which is responsible for mitogenesis, cell division. So MAP kinases are initially found that they are very important, having role in cell proliferation, cell division, cell proliferation, cell growth. Understood? So that way, this MAP kinases forms the central uh, pathways of signal transduction in the cell, and these MAP kinases are different types of isoform that it has got. Major are ERK pathway, ERK, extracellular signal regulated kinase. ERK pathways are responsible for cell proliferation, then P38 and SAPK. SAPK, P38 or SAPK. P38 or SAPK means stress activated protein kinase. Then another one is JNK. So these three are major constituents of MAP kinase pathways. So all these three are what I said, they are nothing but isoforms of MAP kinase, altogether known as MAP kinases. So P38 and JNK pathways are responsible for apoptosis. Okay, so that pathway is connected through this EPAC, and for this EPAC, CAMP is the upstream signaling molecule. So that is what we discussed, and this is the notes for the same. CAMP can act on effectors with specific CAMP binding sites and are regulated by CAMP via allosteric mechanisms. So formation of adenylyl cyclase. So mainly this mechanism is first one you have seen here. This is formation, this is degradation, then targets. So that's way you can explain this pathway. formation, degradation, and targets. So formation, as already we discussed, this is transmembrane. This adenylyl cyclase is present in the membrane. Earlier we discussed. Okay, you also know different types of adenylyl cyclases. Correct. If you forgotten, you have to again, go through. So adenylyl cyclase are membrane-bound enzymes and also soluble adenylyl cyclase. Two types are there. So these are responsible for CAMP formation, and transmembrane adenylyl cyclase are mainly regulated by G alpha, G beta gamma. So this is the part of your heterotrimeric G proteins which are associated with G protein coupled receptors, as well calcium ions. Understood? So transmembrane adenylyl cyclases are regulated through G protein coupled receptor mediated signaling, whereas soluble adenylyl cyclases, which are present in the cytosol, these are regulated by bicarbonate and calcium ions. So calcium is common here. Both GPCR and soluble adenylyl cyclase mediated signalings are through calcium. Understood? But these are insensitive to heterotrimeric G protein. So soluble adenylyl cyclases, they are activated by the bicarbonate and calcium, but they are not activated by G protein coupled receptors in the cell. That is the main source for conversion of ATP to CAMP in the cell. Then degradation, that is by phosphodiesterases. There are 11 families of phosphodiesterases that are expressed from 20 genes have been identified so far. And these different PDEs include CAMP dependent PDEs, CGMP dependent PDEs. In the last slide we have discussed, PDE is same for both CAMP and CGMP. So accordingly, these PDEs can be classified as CAMP dependent, which will work on only CAMP, CGMP dependent, which will work on only CGMP, and there are dual specificity phosphodiesterases. That is, they act both on CAMP as well as on CGMP. That is about phosphodiesterase. So mainly phosphodiesterases are responsible for converting CAMP to AMP. So breakdown of CAMP is because of the phosphodiesterase. So these are under the control of now PDE's action. These are enzymes, right? So their action is mainly controlled by calcium and calmodulin and by protein phosphorylation. So two things will control this PDE activity. One is calcium calmodulin and protein phosphorylations. So these PDEs and adenylyl cyclase are regulating the CAMP, which in turn mediate different cellular effects that we discussed in the earlier slide. That's how these two enzymes are important drug targets now. Many inhibitors, those are the drugs, are used to control the activity of the CAMP-mediated signal transduction in the cell. Understood why we need to study the mechanism? So if you know the mechanism, then you know CAMP is very important and this is formed by the action of these two enzymes. So if you control the activities, if CAMP is more, which you don't need CAMP signaling, then use inhibitors for adenylyl cyclase. So that inhibitors of adenylyl cyclase are your drugs. Understood now how drug discovery will work? So that's the way. Next, as we discussed in the pathway, if you recollect, targets. So we discussed that targets, there are three important targets, right? In the pathway, there are three important targets. One is EPAC, this is nothing but GF protein, then protein kinase G, and the channels, ion channels. So CAMP targets, the gated ion channels, these are under the control of CAMP. Gated ion channels, cation channels, upon its binding to cytoplasmic structural element of these ion channels, when CAMP binds to these ion channels, channel will open under the CAMP effect. So this is seen in case of our perception of the smell perception, olfaction. And this in mammals is because of the CAMP regulated calcium passage through the cation channel. So smell is based on CAMP, second messenger, and now you can see how much fast in the cells that the signal transductions are working. How much time you need to tell me, like what smell you are getting? No, see, these are all mediated by the CAMP, and when they bind to calcium cation channels, and also you need to appreciate here the role of calcium. Okay, so calcium has got very important role even in the nervous system and even in the structural uh, functions of the body. Okay, so that is about the CAMP target, that is cation channels. Another major target is protein kinase A. This already you know. So protein kinase A, can you recollect protein kinase A structure? That is heterotetramer. So two regulatory and two catalytic sites are there, and the CAMP has got four binding sites on two regulatory and two catalytic. After its binding, the catalytic units will become freed, right? Even in FRET we have discussed. So it will become individual catalytic subunits which are ready to activate the substrates. And regulatory will be as a dimer. So the majority of CAMP effects are mediated by protein kinase A. CAMP binding to pKa relieves autoinhibition. This point we discussed. And this allows pKa to phosphorylate the substrate. So this is a protein kinase A. These are serine threonine protein kinases. So they are responsible for the substrate to phosphorylate on serine threonine residues. So upon 4-CAMP, this you know, right? 4-CAMP molecules are binding to pKa, that is 2 regulatory and 2 catalytic. After its binding, it undergoes changes in conformation and separates as R2 dimer and free molecules of 2 catalytic subunits. And specificity and selection of the substrate by pKa signaling. So in pKa signaling, pKa functions is mainly dependent on, we also discussed about this, AKAP. What is AKAP? Can anybody tell what is AKAP? Already we discussed. These are anchoring proteins. So they are localized in the cell in different parts, and mainly pKa functions are mediated through this AKAP anchoring proteins. Okay. So that is about the protein kinase A as a substrate of CA. Then another one which we discussed is EPAC. So tomorrow you need to tell what is the abbreviation for EPAC. Basically EPAC proteins are guanine nucleotide exchange factor, GFs. GFs you know already, right? GFs are opposed by what are the other proteins? Okay. So GFs are responsible for what function you know. So CAMP can also activate these GF proteins known as EPAC, which is a small GTPase RAP1, and it's a transcription factor. This is a transcription factor. Okay. So the binding of CAMP to the multi-domain EPAC protein causes a conformational change leading to autoinhibition, same like in protein kinase A. Once CAMP binds, it releases autoinhibition so that it becomes activated, and this leads to increased exchange activity towards RAP1 and RAP1 activation. So that way, RAP activation is taking place. So activation of RAP by EPAC links CAMP signaling. So through RAP, this is related to MAP kinase signal transduction pathway. EPAC activation, release of autoinhibition, then RAP1 through RAP1, RAF, MEK kinase, MAP kinase. This you know, right? MEK kinase, MEK and MAP kinase. That is MAP kinase pathway. So with that pathway, this EPAC is linked. So EPAC role have been found in diverse cellular. So this particular pathway is in insulin secretion. Insulin, you know, so its secretion is mediated through this signaling pathway, and also cardiac contraction. Heart contraction is also related to this particular pathway in the cells. Understood all of you? Any questions, clarifications you require? So if you don't require any clarifications, then that is all about CAMP. Then we are going to discuss about CGMP. CGMP is like CAMP, and we discussed that its formation is by guanylyl cyclase. CAMP formation is by adenylyl cyclase, correct? And degradation is by PDs, phosphodiesterase. Same way here PDs will act. PDs we discussed three types: CAMP dependent, CGMP dependent, and DL specific phosphodiesterase. So CGMP, like the CAMP, very important second messenger in the cell, and it's produced from GTP. There it is from ATP by the action of guanylyl cyclase. CAMP formation is by adenylyl cyclase. So many targets of cGMP contains cyclic nucleotide recognition molecule termed. So cGMP effectors are having one particular domain, and that is known as GAF, not GF. This is GAF. What is GAF? GMP adenylyl cyclase FHLA domain. Okay. So that is the word from where GFA is taken. So GFA domain present on particular protein means that can bound with cGMP. So it was thought that this GAF can bind to cGMP earlier, but now they found that so you have adenylyl cyclase also, right? So it can bind even cAMP. So both cAMP and cGMP binding domains in the protein are known as GAF domains, to which cGMP and cAMP are binding. So proteins like non-membrane adenylyl cyclase, guanylyl cyclases, and phosphodiesterases. So these are the targets of cGMP. So cGMP binds to and activates cGMP dependent protein kinases, ion channels, and some phosphodiesterases. So the same way like cAMP. So the enzymes which are responsible for formation of GMP are guanylyl cyclases, and these are the enzymes that convert GTP to cGMP. So through adenylyl cyclase and guanylyl cyclase functions, in similar way, these two enzymes are same in its function, but their structure and mechanisms of these two enzymes are different. Mechanism of its activation of guanylyl cyclase is different and adenylyl cyclase is different. So these are classified guanylyl cyclase into two types, same like cAMP. Receptor guanylyl cyclase, that is something like non-polar, and polar means soluble guanylyl cyclases. So soluble guanylyl cyclases are present in the cytoplasm, and these are localized uh, enzymes with heme groups. They are having heme group. And these soluble guanylyl cyclases basically activated by another second messenger in the depiction we discussed. No is nitric oxide. No trick, nitric. So it is nitric oxide. So this nitric oxide is another second messenger responsible for activation of soluble guanylyl cyclases. So whereas the receptor guanylyl cyclases are associated with the membrane, so they are transmembrane segment is present. And these receptor guanylyl cyclases are directly regulated by extracellular ligands and are referred as guanylyl cyclase receptors. Okay, so that way these two different guanylyl cyclases are under the control. Then next one, what is the target of cGMP? So like cAMP, cGMP has got three targets. One is a common target already we discussed, that is cation channels. Cation channels are the common target for cAMP and cGMP. Then main important target is PKG. Up to PKC we discussed already. Now here it is PKG you are coming across. Then another one is cAMP specific phosphodiesterase. cAMP specific phosphodiesterase means phosphodiesterase which acts on cAMP and responsible for degradation of cAMP. So you can see this as a crosstalk. So GMP signaling is in crosstalk with cAMP signal transduction. GMP is controlling the levels of cAMP by activating the phosphodiesterase. Understood the relation? So the major target of this cGMP is cGMP dependent protein kinases, These are also known as C GCase or protein kinase G PKG. Different protein kinases we discussed in earlier class. There we discussed about another classification of protein kinase G. So these are activated by binding of cGMP and they have structural elements similar to protein kinase A. Protein kinase structural elements, you know, R2C2, correct? Two regulatory and two catalytic. But in case of PKG, in contrast with that of the pKa, there is both regulatory and catalytic functions are in one domain. So only one domain is present in case of pKg that have both the domains, that is catalytic and regulatory function. So it acts as a homodimer. It is present as a homodimer and binding of cGMP to regulatory domain relieves the autoinhibition. This mechanism is same, relieving the autoinhibition of N-terminus and allows the phosphorylation of substrate proteins. But here, difference from pKa, pKg is it exists as a homodimer. Only one type of dimer is present, and there it is R2C2. But here it is same, having two domains. Two domains means two motifs in the same the peptide it is having. That is, one is regulatory and one is catalytic. Understood, all of you? So PKG importantly exerts its function on smooth muscles by acting calcium channels and myosin specific protein phosphatases. So this PKG, CGMP PKG signal transduction is controlling smooth muscle relaxation. See here, these are the different physiological functions of this pathway. So smooth muscle relaxation like vasculature of GI tract, bladder, contraction, peristalsis, you know, like you are eating the food, that is nothing but peristalsis. Anti-peristalsis also, you know, that is nothing but when I tell immediately to write exam, then you will start anti-peristalsis, that is vomiting. So these are all the functions under the control of this PKG signaling. Likewise, even platelet aggregation, that is in the wound formation. Okay, renin lipase release. Renin is a kidney which controls the hypertension, blood pressure. So renin from kidney, that is also under the control of this PKG signaling. Then intestinal secretions, you know what are all the secretions in digestion from intestine. Even in learning and memory process also, the signal transduction has been found to have the role. So this is important effector of GMP. Along with that, cation channels also another common targets of cGMP along with CAMP. So this is all. Now we completed CAMP and cGMP under second messengers. So all of you please go through from your side, otherwise you will be confused. Okay, that confusion you should not. You should appreciate each pathway you have to practice, then what its function of the cell within the cell. That is for your enthusiasm. Okay. So any questions, any clarifications, all of you please unmute and tell me you have understood and followed.
Yes,
Yes,
sir.
yes, sir.
Okay, today I will be sharing all my PPT slides with you all. So you can, if you have not started preparing notes, you know, you finish that task also. So see you then. We'll meet tomorrow again.